months.
Yes. We had 3 different
How
mouse.
big say I'm on?
In that couple weeks that we had that one intern guy, that was really good. We actually looked at the Bible. That was the only time I've ever looked at the Bible. I also got different personalities. Oh, she's fine. Me too. I can watch it. She's really funny. I know.
Okay, well, good morning. It's good to be back. Anyways, game plan this week, and I think I posted it on Friday, and hopefully you looked at Google Classroom. I should have told you to. I'll just re-lecture Chapter 13 because there's a few things that guy kind of skipped over and didn't really go into depth. So I'll re-lecture 13. Tomorrow we'll extract our DNA. We'll lecture on Chapter 14, which is about DNA. Wednesday is set up to be a review day because I figured, I probably need a day to kind of digest everything. And then plus Wednesday, there's no sporting activities, so then you can study for your exam, which will be on Thursday. So that was another reason why I set it to Thursday. So, yeah. So Chapter 13 was about chromosomes and how we can map chromosomes and how we kind of figured out that chromosomes do carry genes. So the story starts off with Morgan and his fruit flies. So this was the early 1900s, and he was studying a mutant male fly that had white eyes instead of the wild type, which is red. And so he wanted to figure out if white eyes were dominant. So what he did is he took that white-eyed mutant fly and crossed it with a red-eyed female, and all the F1 crosses had red eyes, which stated that, okay, red eyes are dominant to white, and so that's what he concluded. So then he took those red-eyed fruit flies from that cross, and he crossed them together, and he noticed that only 18% had white eyes. And so the ratio was greater than this, it did fit that Mendel's ratio of 75% to 25%. And another key thing that stuck out, not only the 18% had white flies, but that the only ones that had white eyes were males. And so this was really puzzling to Morgan. So then he took these white-eyed males and he crossed it with the female F1 progeny, and he got an even more crazier ratio, a 1-to-1 to 1-to-1 ratio, where you had white-eyed to red-eyed flies of both sexes. So there were some white-eyed females in this group, and they were viable, which means that they could reproduce. And so he wanted to figure out what was going on here with the chromosomes. So then he said, okay, because it showed up in males first, maybe it's tied to the sex chromosome. So the gene for eye color does lie on an X chromosome, the sex chromosome. So like for us, we're XX, girls are XX, boys are XY. And so this white-eyed trait does reside on the sex-linked chromosome. So his results provided clear evidence that genes do reside on chromosomes because of that sex chromosome. And then, yes, there was a slide showing that the structure and number of sex chromosomes do vary between species. He mentioned CW for birds and ZZ with grasshoppers is XXXO, and with honeybees it's not, there is no sex chromosome, it's how many chromosomes you have. Do you have one set or pairs of it, so diploid or haploid? But in humans, the Y chromosome does determine maleness. And this is another reason why I want to go over it, because he didn't talk about the SRY gene that is found on the Y chromosome. So on the X sex chromosome of humans, there's over like 1,000 genes. It's a lot bigger than the Y chromosome. The Y chromosome only has like 100 genes. And all of those genes pertain to maleness. There's a specific gene on the Y chromosome, the SRY gene, that is actually responsible for the production of the genitalia and the secondary sex organs that pertain to males. And so if you have that one Y chromosome, you are normally male. So you could be XXY, you'll still be male even though you have two X chromosomes. So this diagram kind of talks about sex determination in mammals. The default is to be female. And The only reason why you would be male is if you have that Y because of that SRY gene. And I cannot stress this enough. I'm pretty sure I have multiple choice on the exam that states what gene is responsible for maleness or something like that, and it is the SRY gene. So if you don't have the SRY gene, that hormone or that protein does not get made, and therefore you turn out to be female. So there are some exceptions, okay, to the rule provided for the mechanism of sex determinant. Sometimes the Y chromosome, a movement of it can cause XX individual to develop as male. We did talk a little bit about genetic disorders. If you do have a mutation in the SRY gene, then you could turn out to be female because that gene is malfunctions. It doesn't work properly anymore. So then, you turn out to be female. Sometimes in vertebrates, okay, it's not determined by sex chromosome. It's determined by your environmental factors like fish and reptiles. So I'm sure you've heard of sea turtles, right? How they're determined by temperature. If they're above a certain temperature, I think I wrote it down here.
Does that mean if a female lays like multiple eggs in the same spot, there won't be any difference?
Well, so this is, I think I made a little note here. It says the sex of most turtles, alligators, and crocodiles is determined after fertilization. The temperature of the developing eggs is what decides whether the offspring will be male or female. It's called temperature-dependent sex determination, or TSD. Research shows that a turtle's eggs incubate below 81.86 Fahrenheit. The turtle hatchlings will be male. So it has to be below 81.86 females. If it's above 81.8 Fahrenheit, the hatchlings will be female. Temperatures that fluctuate between the two extremes will provide a mix. No, not for one individual. I'm just saying the eggs. The eggs. Yeah. So that's how you get male and female. But if it was pretty much the same throughout, then it's most likely that they would all be, female. So that's the problem right now with the climate change. Is that our temperatures are getting warmer, so we're seeing more females and less males, and so it could be endangered. Well, yes and no, because that one female, she's gonna mate.
You just get made with like 7.
Right, but if you have few males and a bunch of females, not all those females will mate. So yes and no.
I just figured the male would hit up more than one.
Well, that's the hope, but yeah. So we do have some genetic disorders that are sex linkage. Red-green color blindness, more common in males, but it can pertain to females. Hemophilia is also sex linked, and so basically there's a protein on the X chromosome that doesn't allow your blood to clot, so you can fall down and get a little bruise and bleed to death internally from it and die. So that's what hemophilia is. Okay, another reason why I wanted to re-lecture is he mentioned that females could be mosaic, and I think that's not true. I think we are mosaics. Girls, we are mosaics. And so we do have 2 X chromosomes, but one of them does get turned off. So we call this dosage compensation, which means one gets turned off so that way you get equal amount of expression from the sex chromosomes, so you don't get an overload of it. And so one, shuts down and becomes a bar body and is inactivated, and the other one is the one that is activated, and it's totally random which one gets turned off. So it does it's totally random from cell to cell. If a woman is heterozygous for a sex-linked trait, some of the cells will express that one allele, and the other cells will not. The one that is inactivated gets highly condensed, which means it shrinks down and gets super coiled up, so proteins can't latch onto it and read it, and it's just visible as a dark stained bar body. And then it hangs out near the nuclear membrane of it. So here's a zygote where we both have our X chromosomes, and then after fertilization, the cells divide mitotically very rapidly, and then after that mitosis, some cells shut off one X chromosome, the other cells shut off the other. And so it's totally random what happens. And then these cells go through more mitosis. And so, yes, females, we are mosaics, or some cells express that one allele and the other cell does not.
So bar body would only be in a female.
Yes. Yep. So we are genetic mosaics, and so in art, you guys know what a mosaic is, like broken little pieces. A lot of times you find it in pottery, but it usually has a picture of these broken little pieces. And so females that are heterozygous for the X chromosome, we are genetic mosaics. Some cells express that allele, some cells don't. Well, one example in vertebrae is calico cats. Females have a patch distribution, dark fur, orange fur, white fur. And it's all because of that heterozygous allele, which one is being expressed. And I think this is another reason why, I was looking at my hair, I know this is weird. I was just looking at my hair, and I actually have dark red hair, a few strands, one every couple hundred. It's what the heck? My mom has red hair, so that's just. So here, males are usually solid color because they only have the one X that gets expressed, so they're either orange or black. Females, there is the possibility that both X's are little b, resulting in black, or capital B, resulting in orange. Or if they're heterozygous, then you get that patchy colorization. Okay, then he goes on to mention a little bit about not all organelles follow chromosomal theory of inheritance. We do have organelles that have their own set of DNA, mitochondria, and if you were a plant, chloroplast. It's all because of that endosymbiosis theory, where at one point they were probably a prokaryote that got engulfed by a eukaryotic cell, and then they started to live in symbiosis with each other, and now we just have mitochondria and chloroplasts that live in harmony. But Ruth Sager examined these non-Mendelian inheritance with organelles and came up, proved that, mitochondria and chloroplasts, they don't follow the rules of Mendel's genetics. In fact, we inherit them from our female parents, so they are maternal inheritance. So you get all of your mitochondria from your mom. You have the same mitochondria as your mom and as your grandma. So the zygote forms, it receives an equal contribution of nuclear genome, which is DNA from each parent, but all of your mitochondria come from the egg cell. The sperm just contributes the DNA, doesn't contribute any other organelle. And so as the zygote divides, so does that original mitochondria that you got from your mom. So when we look at DNA, you're an equal mixture of both mom and dad to get you, but for mitochondria, you're only looking at your mom, which is maternal inheritance. And we'll actually look at a case study later on, I think this semester or next semester, where you will you'll have a case study about Anastasia, whether or not she's dead or alive, and you have to look at mitochondrial DNA that they've collected. And then you'll also look at King Tut and his family tree and how messed up his family was. Again, yep, that's pretty gross. I mean, and then how he died is another thing.
Did he get beheaded?
No, he was not beheaded, but he was malformed and just genetically messed up, and because his dad married his sister too, and so it's what is going on? So it's interesting to learn more about that, just because of trace mitochondrial DNA, who's related to who. So we call this maternal inheritance. There is a disease associated with maternal inheritance and faulty mitochondria. It's called LHON, Leber's hereditary optic neuropathy. And basically what's happening here is that your NADH dehydrogenase enzyme doesn't work right. And if you remember in the ETC, this is the first place where electrons get dumped off. Okay, so if this is really faulty, those electrons never get into the ETC. And then you have the FADH2 that goes to the FADH2 dehydrogenase kind of complex area. So some ATC does get made, but it's not the full amount that you could. So it does reduce your ATP production, And that's why your nerve cells start to go first, because they're the ones that need energy all the time. I mean, you're constantly using your neurons to do everything. Okay, so far with Mendelian characters, yeah, there's genes located on chromosomes. We know that they line up randomly in meiosis through independent assortment. And we've also looked at 7 traits of Mendel as either or. And yes, they all show an independent assortment with one another. Mendel got really lucky looking at pea plants, okay? But the ones on the same chromosome, because there were some traits that were on the same chromosome in Mendel's, that should not have behaved as they did on different chromosomes. And so we have to look at what happens when genes are on the same chromosome and this event of crossing over, because we know that chromosomes cross over. So crossing over during meiosis does increase genetic variability. And so like here, if I have all homozygous dominant to homozygous recessive and we cross over, then all of a sudden I get recombinant combination of alleles that can get expressed. And this is what happened with some traits that scientists were looking at. They're like, wait, how is this even possible?
The alleles like A and B.
Yep. So alleles are always designated by
The
capital E,
chromosomes are those.
the colored things.
There.
Yep.
Now what are the genes?
The genes? No, this is gene/allele. So the alleles, so this is gene A. I don't know what A stands for.
Okay.
Could stand for, eye color or ability to roll your tongue, and this is dominant or recessive. So genetic recombination means that alleles do exchange. And if we look at a dihybrid cross with 2 true breeding parents that differ in 2 traits, we should get a double heterozygous F1 progeny. Okay? And it goes on to say that if genes for 2 traits on a single chromosome, and during meiosis, we could expect that those alleles for both locations should segregate or separate together and produce gametes that resemble the 2 parental types. But they were discovering that was not the case. Okay? If crossover occurs, then each homolog would have 1 allele from each parent, and you would get a mixture of the parents. And this is what we call the recombinant gametes. So if I go back a slide to kind of show that diagram again. Okay, here are the parents, right? And we have crossing over. This strand is the same as the parent. This side, okay, of the sister chromatid is still the same as the parent. But the 2 middle ones are not because of crossing over. So we call these the recombinant chromosomes because eventually they'll separate into the gametes. Okay? And these guys are the non-recombinant because they are identical to the parents.
And the chromatids, is that like just the one side?
Yep, so this is a chromatid, this is a chromatid. They're sister chromatids, this is a chromosome. These are homologous chromosomes because they carry the same genes at the same spot.
Homolog.
Yep, homolog. Yep. So lots of vocab here. Yep. And then because these are homologous chromosomes, that means one came from mom, one came from dad. Okay. Now I want to go back to the fruit flies. Morgan again provided evidence of these recombinant chromosomes because he got that 1-to-1-to-1 ratio, that mixture, and he's like, well, that should not have happened with Mendel's genetics. And so that was another piece of evidence that showed that crossing over occurs, and we got this recombinant genotype that were completely different from the parents. Barbara McClintock also, with again Harry Crichton, looked at corn, maize, and she also provided physical exchange of genetic material.
Is that where they got Creighton University?
I don't know. So I want to go back to Morgan. Okay, here he had a wild type fruit fly. Okay, so this is normal. This is the female, and then he had a male that had, in this case,
Would Logan Young, Brody Morrow, and Parker Bartel come to the office, please? Logan Young, Brody Morrow, and Parker Bartel.
it's a weird combo. Black with vestigial wings. So black is the mutant color. The little folded up wings is also mutant. Okay, and we get 100% this wild type, this heterozygous, big B plus, B with no plus, the VG plus, the VG with no plus. So that's what this arrow means, is that 100% were heterozygous. That makes sense. But then they crossed this, okay, again, with the double mutant, and they got this kind of 1-to-1-to-1 ratio, where we had 25% look like the parents, but then we had gray vestigial, which means gray was normal, but the wings were mutant, and we had black, that's the mutant color, and then normal wings, which was parent. So it's a mixture of the parents. They don't look like the parents. They have dominant and mutant phenotypes. Okay, so these are the recombinants. So these were the result of the crossing over chromosomes that had both alleles, dominant and recessive. Okay, the Mays experiment, she noticed that one chromosome in the corn had a little knob and that it was completely different from all the other chromosomes. So she called this a cytological marker that she could follow in the fate of meiosis to figure out what does this chromosome do. And she determined that this gene does determine kernel color and kernel texture. So this longer chromosome had the knob, and it did carry a dominant color allele, which we'll denote by a capital C, and then this waxy allele, which we'll denote as WX. And so she took the longer chromosome, and so big C, WX, and crossed it with two little C's and a capital W and X, so to be the opposite of waxy. I don't know if that would be smooth, I guess. No, it'd be rough. And all the progeny were colored and starchy. I guess starchy is the opposite of waxy. And so, yes, they were heterozygous for both loci. And then she did a test cross, and she realized that all the progeny exhibited recombinant phenotypes, which means that they did not resemble the parents. They had some features of the parents, and it was all because of this chromosomal marker that she was following. She could prove that it did. Now, recombinants are super important in mapping out where genes are in chromosomes. And so in the lecture last Thursday, he did mention a little bit of Alfred Sturtevant, a student of Morgan, who I guess one night blew off his homework and wanted to figure this out, and he did. And he reasoned that the frequency of recombination, how many recombinants you got in a progeny, was the measure of the genetic distance. And so, yeah, chromosomes, you can figure out, crossing over the distance between each genes. And so as physical distance on the chromosomes increase, so does the probability of recombination, which makes sense. So if you look at the recombinant number or the recombination frequency, you can in turn map out where those genes are on that chromosome. So they named it after Morgan, not Sturtevant. They call it a centimorgan for a map unit. So like if they said, if I had 8% recombinants between this body color and eye color, then I know that they're 8 map units apart. And if I got vestigial wings with eye color that occurred 9% in the progeny, I know that they're 9% apart. But then if I looked at wing size and body color and it was 17%, well, it's kind of like using math, where you can place them in order that fits the percentages. So you can construct maps. It's kind of like working backwards. You take a look at your progeny, you figure out the recombinant percentages, and then from there, literally draw a line. So like, for example, if I was looking at A and B, and it happens like, we'll say 10% of the time, and I was looking at B and C, and it happened 5% of the time, and A and C, we'll say it was 15% of the time, you can map this out. So I know on my chromosome, A and B are 10%, right? And then it says B and C, well, either C can go here, or it can go here. But if I look at A to C, it's got to be 15%. Well, that wouldn't make sense because it would only be 5%, right? So then I know C is here. So there's the 5, and then there's the 15. So that's actually what scientists do. they can do this. But now we have fancy machines that can tell you exactly where it's at. But in college, if you have to take another biology class, I'm sure you'll have to do this. You'll have to look at the percentages and map it out. And it's kind of like just doing a math problem. I remember doing this in elementary school. If so-and-so is so far away, where are they seated? So here's actual data from Morgan and students who work, where they got 405 parental, 405 15, so they look exactly like the parents. But then we had these recombinants, 92, 88, and we had 1,000 progeny. So then you can figure out the map distance between these two loci by figuring out the percentage of how much it could. So. Now, chromosomes don't just cross over once, they can cross over multiple times. So like, here's a chromosome, that's really bad. They can cross over here, and if I were to extend it, they can cross over at the top too. So this does take into account that, yeah, chromosomes can cross over multiple times. What happens when you have more than one recombination event? Well, if you undergo 2 crossovers between, then the parental combination is just restored. They're just on different chromosomes. So I don't know if I think I have to go back to that chromosomes picture. So they're saying that, okay, if we crossed over here and then let's say that these crossed over again, well now it would just be all the little letters on this side and all the capital letters are on this side. That's what they're saying. So then you've just restored the parental.
So then you're saying they'd like flip and then flip back.
Right. So if homologs undergo 2 crossovers between the loci, that parental combination is restored. So. Okay, you will never get a recombination frequency of above 0.5. Okay, because then past that, that means it's restored back to the parental genotypes. So they just say 50% is the cutoff. Okay, the last kind of bit here is talking about human genetic disorders. So yeah, there are some genetic disorders that are the result of, a mutation in a protein. So sickle cell anemia is a single point mutation where it should be A to T, but then the A gets mutated over here to a T. So instead of glutamate being produced for the normal protein, valine gets produced. And now all of your red blood cells are sickle shape. I shouldn't say all of them, but most of them are. Non-destruction means that you changed your chromosome number because during meiosis they didn't separate properly. And so if you have a plus or a minus chromosomes, we call it aneuploidy. If you lose a chromosome, most of the time you don't come into it the fetus aborts. But if it's the sex chromosome, depending on which chromosome gets shut off, it may come to term.
If the Y gets shut off, then you can still survive.
Right, so you'd be female. So mononomics just means you lose a copy. Like I said, you usually don't survive, but if it's a sex chromosome, yeah, you got a chance. Trisonomics means that you have a triple number. Few cases where they do not survive, but on the smaller chromosomes, which are closer to 23, like Down syndrome, it will come to term.
Isn't like trisomy 18 a thing too?
Mm-hmm.
So could you have trisomy 18 and 21 for a total of 48?
Ooh, I don't know.
Plus the sex
chromosome. That would be very rare.
Plus the sex chromosome for 49.
That'd be very.
Go on board.
So yeah, you can have non-disjunction with sex chromosomes, and so I think the guy last Thursday did a great job going over each one of these. But XX, female, you get sometimes. What am I trying to say here? Female with 1 functional action and 2 bar bodies, so XXX, so triple X syndrome. If the XX combines with the Y, then you get Klinefelter syndrome. If one gamete doesn't have the O, but the Y does, then it's non-viable, it fails, it aborts because you need that X chromosome and it's got over 1,000 genes that are needed. If the O combines with an X, then you get a sterile female. We call this Turner's syndrome. Sometimes the Y chromosome fails to properly separate in meiosis, so you can get a formation of a YY gamete. If the YY combines with an X, you get XYY. It does develop into fertile males. They have a normal appearance. We call it Jacob's syndrome. So yeah, you could be XYY and not even know it. Okay, so that's chapter 13.
There's a guy who only has 44 chromosomes.
Really?
And he survived. He has none on 15.
Wow.